Thank you, Kim. I just wanted to echo something that Kim said at the start of her talk, which is that this has been a really challenging year for our community, and that we've lost some of our most distinguished and influential colleagues. And sadly, this year's winner of the Herpetologists League's distinguished herpetologist, Ray Semlitsch, passed away this year. And fortunately, we do have some members of Ray's family, some members from his lab, and Jim Spotila here to speak about Ray's accomplishments and the wonderful life that he lived. So we'll have Jim come and tell us about Ray.
Thank you very much. It's always a wonderful time to present the Distinguished Herpetologist Award from the Herpetologists League. Today it's bittersweet because it's for Ray Semlitsch, who passed away in June from acute myelolytic leukemia, if I remember correctly, and was unable to make today's presentation. But his son John, I'd like to come up. If you come up, John, I'd like to present this to you in Ray's memory.
I just wanted to let you all know that my father really would have loved to be here today, and instead I am, and I'm very grateful for this opportunity. I'm very proud to be here and accept this one. So thank you all very much, and have a great time, obviously.
Last time I talked to Ray, when we were still hoping that he was going to get better, he said if he didn't get better, but if he wasn't well enough to attend, he asked if I would give a presentation for him. So I put together a presentation about Ray, which I'd like to present to you. The title of his talk was 40 Years of Snakes and Salamanders, Experimental Approaches to Herpetology. Here's a picture of Ray in his natural habitat, which is the forest. Here he is with his almost lifelong friend and colleague, his good buddy Whit Gibbons, who received the award last year. This is when he got his pre-distinguished herpetologist award in 2011, the Fitch Award. We can go by Ray Semlitsch by the numbers, and I'm not going to read that. You could read that. Published a lot of papers and book chapters, books, distinguished journals such as Science and Herpetologica. He's been cited over 13,000 times, etc. But Ray's legacy and his career as a herpetologist and ecologist was more than numbers. It was his total impact on both the science and the people. And so today, I'm going to try and do what Ray would do, and that is give a talk to the younger people in the audience. The older people like myself or Tracy, or we're a hopeless anyway. Aaron Bauer, we're all way past the hill. So there's no use talking to ourselves. But I'm going to try and give a talk that might show what it is that he did and what might be some lessons. So what are the lessons learned, as the military likes to say? Lessons learned from a lifetime of herpetology. What do we learn from his legacy? Part of his legacy, this is a picture of his former students at his memorial service in Missouri in June. Well, that's part of his legacy. Going through, to give a talk about what he did with all these papers, you have to go through at least the titles of the papers, And I found many themes in Ray's lifetime work, and here are his themes. From the love of natural world, to mentoring of students, being kind and generous, his incredible organization. He was a humble person, and he had a positive attitude always. Where did this come from? Where did he get this? Well, in the beginning, if we say in the beginning, and from high school in 1968, he joined the United States Marine Corps. And if I remember correctly, there's another ex-Marine in here, if he's in here, Vic Hutchison, who also was a Marine years ago. He did a combat tour in Vietnam, in I Corps, and those of us old enough to remember, that was a place you really didn't want to go. It was up near the DMZ, and you walked around up and down hills trying to find the enemy and kill the enemy. So he received numerous awards, no great medals, but the usual awards for being there. And he came back, and here he is in Buffalo. I think this is probably before he went over to Vietnam, but I remember talking to him, and asked him, well, Ray, did you see any herps? Did you do any herping when you were over there? He said, no, mostly tried to stay alive. Didn't have any time for any of the herps. And he also mentioned that when he came back, this of course has effects on people when you do this sort of thing. That he couldn't sleep in his bed, he slept in the garden, and his parents had to come out and get him to come back in the house. So he had some things to adjust to. He got an honorable discharge in 1972, and using the GI Bill, he went to college. Buffalo State College, where I was a professor, and he came to me early on. He said he had this thing about research. He wanted to do research, and he was an intern at the Buffalo Museum of Science, and they got an alligator skeleton, and he wanted to put it together and get Bio 495 credit, 3 credits for putting together the alligator as his research project. And you hate to chase a student away who you think may have some potential. So I said, Ray, why don't you read this, my salamander paper, my temperature and water balance, and the salamander paper from '72 had just come out recently. Take a look at that and see if that gives you any ideas. Well, he did read it, and he's mentioned that that sort of gave him an idea of what science was about. And so he came along to the Savannah River Ecology Lab on a field trip, class field trip, and he fell in love with herps. He did research as an undergraduate when there was still DOE money now for undergraduate research down there, which was pretty much dried up. And he got a BA with honors. Now, Sorrell's, the Savannah River Ecology Lab, is a herpetologist's delight. It's a herpetologist's delight just like military bases, because you can't go there unless you get a badge. And so you've got 300 square miles to play herpetology, salamanders, frogs, snakes, lizards, whatever. And public can't go there and mess up your experiments. So this is a wonderful place. It was there because of hot water, because of nuclear reactors. We had to make plutonium for our hydrogen bombs, and as a result, we had to put a buffer around them. You'll notice that this is the old picture of Parr Pond, where hot water, and we did a lot of fish studies in those days, and Pond C, and notice Pond B was also not hot anymore because there was this inoperative reactor, which had actually melted down. Didn't hurt anybody, don't worry about it. In the early days, here's Ray with Cindy Scott from Buffalo State. There's Wick Gibbons. You may not recognize him as a young person. And the rest of the crew at the old lab. He did research on snakes and salamanders. And his first paper is very interesting. Semlitsch and Gibbons, 1978, undergraduate work. I don't know if you can see it if you're close enough, but it's a note in Copia, reproductive allocation in the brown water snake. And look at his first sentence. The concept of reproductive effort has played a major role in the development of theories explaining life history patterns. However, the development of theory has outweighed the accumulation of empirical data. This is true then and is still true today in many cases. So that's what he did. He accumulated, he studied, and got those empirical data to test these theories. His first sole author paper came as a side piece of that allocation paper in the Journal of Herpetology in 1979, in which he studied shedding rates with temperature. He required catching lots of snakes, and he allowed me to go out and catch snakes with him. So we go out in the John boat in the swamp, and you go, and there's an overhanging tree, and he says, grab the snake, and I say, where? He says, just grab the middle, because it'll turn around and bite you, and then you grab it. So we switched. I drove the boat. We grabbed the 25 snakes, came back with bloody arms and 25 Natrix type snakes, and a few water moccasins. And one question I had was, Ray, I don't want to grab it if I can't tell if it's a water moccasin, if you can't really see the head and the markings and stuff. He said, don't worry about it. Water moccasins are always coiled up, and the water snakes are spread out. But he was careful enough. He wasn't foolhardy. Using snake sticks, I'd drive the boat up on the shore, and he'd run out and grab the water moccasin. So that was great fun for an ex-Marine, I guess. Here's another view, and here are here's the data, quantitative data, shedding at different temperatures. Well, what were the lessons learned from Ray as an undergraduate? He was disciplined. He got that from his training, I guess. He was enthusiastic and focused on learning and success. He always took advantage of opportunities when they came up. My father used to he got me a job in the shop when I was a student, college student, and I came home once and I said, Dad, they want me to work on Saturday. I want to go out and catch salamanders. He says, never turn down overtime because they won't ask you a second time. Ray had the same attitude. If there's an opportunity, seize it. And that's a good lesson. He was willing to fail. He wanted to be the best. Published papers. His master's thesis in 1979, not that much later, with Dick Heighton, on one of the most highly evolved species of Ix and Herps, the Plethodon, the population variation. And his first work with an undergraduate, mentoring an undergraduate, came out in 1980. The Semlitsch approach was quantitative field research, so a typical page for one of his papers, data tables, statistical tables, even, regressions, and here you get the size, numbers of animals of different sizes in different places. He was always quantitative in anything he did. He was also a multitasker. While working at Savannah River Ecology Lab on snakes and salamanders, he also worked on salamanders in the mountains, mentored undergraduates, and developed his style as a researcher. While doing his PhD at the University of Georgia, another 16 papers. Everything from salamanders to turtle snakes and toads. And here's some of the crowd. There's Ray, Whit Gibbons, Steve Moriali, that's Judy Green, who did most of the work for all these guys, and I think Joe Schumacher. In 1983, life history and ecology was an obvious focus for Ray, and it remained the same throughout his research. Postdoc at Duke, another 6 papers, and here, first work with a guy named Joe Peschman as an undergraduate, his second undergraduate, on patterns of migratory activity in salamanders. Got involved in long-term studies. The long-term studies on the Savannah River plant spanned 44 years. Peschman is the lead author on this 91 paper, which made the cover of Science, in which human impacts on natural fluctuations were important. As assistant professor, he was at the University of Memphis and then Zurich. I have no idea how he remained so productive. This guy's always turning out papers. I never could figure that out. But besides herps, he studied other things like caterpillars. He started his work on metamorphosis there. He studied the germination success in acorns. And started working in a hybridogenetic Rana esculenta. And throughout this process, starting at Sorel and going through Memphis and Zurich, he used this approach of mesocosms, cattle tanks as mesocosms, to study frog tadpoles under different densities. So he could create different ecosystems in these tanks and thus study interactions of these animals. Then he went on to Missouri. Here he branched out to study female cloacal anatomy of salamanders, of course. He came under the influence of some, strange people, Dick Sevier and Trout. These are people who probably are on somebody's list. Today they would be on one of these lists of who lives in your neighborhood list, sort of thing. But many people in this room are probably on those lists anyway. I know Tracy and I are on lists like that. He also began to study effects of pesticides, so he's branching out into pesticides effects on amphibians, which he started in Zurich, his first study on the effects of tin compounds, not just on survival, but on swimming and feeding and sublethal effects. Another theme that he developed. Then he worked on these long-range studies, 44-year studies of counting amphibians and reptiles. This is on the Savannah River plant. And this was one of these, Whit Gibbons wanted to have the most number of people on a paper. So he's the first author, but it's a rogues gallery. Vincent Burke, I always wondered what ever happened to him in science. There's Jeff Lovitch, so you know you're in trouble right away. Ray Semlitsch, you can see that list of people on here. Even Mike Dorcas and Justin Condon. But the whole host of characters that for 44 years are following these animals. And what do you learn from that? The famous Rainbow Bay study for 18 years, you get lots of additional species added of amphibians and reptiles over the first 5 or 6 years. But by the time you get up to 400,000 individuals, you start getting some quantitative data. So at Missouri, all these ideas came together in his lab, and he had students studying many different aspects. This paper by Alison Welch on call duration got him into sort of evolutionary ecology, which was frogs that call longer and stronger have better offspring. So it published in Science. That's great. When you publish in Science, then you get somebody from a medical school writing in and saying, well, all you really did is devise a handy-dandy kitchen device, which is, was a strange comment, but it was something to the effect of, this is a device that does everything. Opens cans, beer bottles, shapes potatoes. It has a job, you can buy it on TV for $5.95. So that's all this was simply many things happening at once. Their response, of course, tried to illustrate that we're talking about selection, individual selection, and in particular genotypes that are tied together. Well, what's the greatest sort of joy for the professor? Fame and happiness to the professor comes from the success of their students. And in this case, Michelle Boone did additional studies on insecticides and interactions with pond drying on amphibians. Very successful, and she went on to work at USGS and publishing papers on her own. And many of Ray's students are off on their own doing science. And I think that's one of the things that really made him very pleased. He was also willing to learn from others. So Vince Burke came up with this idea that there should be sort of boundaries and buffer zones around turtle nests. And Ray picked up on that and published several papers on the importance of small isolated wetlands. This 1998 paper in Conservation Biology emphasized that. So it's a very simple concept. You have a wetland, then you have a sort of a wet area, you have an area here which is the aquatic buffer and the terrestrial buffer, and that gives you the core habitat you have to protect. Now this seems pretty straightforward, but it wasn't for the public at large, and following on the park idea, it's a simple idea with major implications. For example, the regulations in New Jersey for wetlands are now based on this concept. Instead of protecting 25 meters around streams in the Pine Barrens, now it's a bigger area because of his work, because of that work. And the protection of the northern pine snake in New Jersey is taking on a similar bent. He also did outreach to the rest of the world. You write a nice paper, it's great, all 14 colleagues read it, and then it gets filed away into your promotion file. But when you reach out in environmental management, National Wetlands Institute, or the environmental law, newsletters, it starts to influence policy. But he continued to back that up with experimental research, landscape resistance, and effects of timber harvest. This is a really nifty experiment that's been done where you take a wetland and you have 4 sections around it. You leave one alone, you cut one and remove the vegetation, you cut another section and leave the fallen trees there, and then another you do a partial cut. Maine, Missouri, and South Carolina. It took 5 times to get that funded through NSF, but it turned out to be a really powerful study methodology. Here's the Maine site with one of those. Who would guess that an abandoned road, 80 years old, would still be affecting salamander abundance in the Appalachians out to 60 meters, 80 years later? At the cycling time of cutting the forest in the southeast, which is about every 80 years, you haven't recovered the salamander population along these roads before you put new roads in to cut the trees again. Finally, wrapping up in 214, we see that he's foraying into policy again. This is in bioscience, and it's a paper on pesticide regulation and the influence of industry. Michelle Boone is now the senior author, along with Christine Bishop, who's worked at the USGS. But it includes people like Joanna Berger from Rutgers, and Christopher Solis, who worked for the EPA, was an undergraduate with me at Drexel, and Ray's on this paper. The point of the paper is that we put 500 million kilograms of pesticides into our environment annually, 1.2 1.5 kilograms for each person in the United States. And the regulations are based on the data provided by the pesticide industry, so there's a little bit of a conflict of interest. And that stirs a lot of dust up in the air. People like Tyrone Hayes have discovered some of that dust out in Berkeley. And so now he's come sort of into policy, most recently. But he's come full circle, but he never gave up the roots he started with. Here's a 2014 paper on the dynamics of ring salamander populations in Missouri, in the Ozarks, published in Herpetologica in 2014. So what he learned as a young man, he practiced as a professor and a scientist. And those themes I started out with, I think if you look through his papers, I tried to catch it quickly in this talk, they carry through. He was always a lot of fun to be with. I think that's maybe Bear's dog from years ago. And this is probably at the Kellogg Biological Station chasing animals. And this is at Rainbow Bay years ago. Notice all the people, but notice Ray. He really seems pretty happy and pretty organized, and people seem to really enjoy working with him, which is the case. So the final lessons, why do you work so hard? Why was he so organized? Why do you have so many students? Why do you mentor them? He did it the reason all of us who are professors do it, because he loved his students. He didn't do the work for him. He challenged them, forced them to work on their own, come up with ideas, and supported them in such a way that they would become successful scientists. Beyond all that, of course, he loved his family best. And that was Ray Semlitsch. And there's Ray in his habitat, and that's why he's Distinguished Herpetologist this year. Thank you very much.